Thank you, everybody, to be there and to be present today. So this is the second video conference in our series. And today, Gaël Recher from the GDR IMABio Steering Committee has gently accepted to moderate this session. So Gaël, if you want maybe...
Yes. Thank you, Virginie, for organizing this. So indeed, today's session will cover the topic related to the application of fluorescence microscopy in studying the relationship between protein structure, dynamics, and function. And this is the occasion to invite both Dusan from the Confocal NL company, who will talk about more the technology, and then on the other side, Erwin Peterman from Amsterdam, Netherlands. So he will talk more about the science and the academic and scientific question related to the use of that technique and how it can be used in the lab. Virginie, maybe you can...
Yes, I try, yeah.
If you have the hand on this, sorry for that. So maybe we can start right away with Dusan, if you are ready, and then we'll have Erwin, and then a short section of question and answer in the end. So the floor is yours.
Thank you. And thank you for organizing. Merci beaucoup d'avoir organisé le webinaire. Et aussi, merci de m'avoir invité à dire quelques mots de solution de microscopie de Confocal NL. Thanks a lot for organizing this and allowing me to say a few words. I'm Dusan Popov-Cheliketic, and I'm the product manager in Confocal NL. Before working in industry, I spent more than a decade in academia, and I'm doing super resolution since 2010. Regarding super resolution, much more of an authority is the second speaker, that is Erwin Peterman. Erwin Peterman will talk after me, and he is the full professor at the Vrije Universiteit Amsterdam. Erwin did his PhD in Amsterdam and at the same university, but then he moved to postdoc in the States, first in Stanford, and he went to a group of William Murner, who is actually Nobel laureate in super resolution microscopy. After that, he came back to Amsterdam, went through all the necessary academic advances to become a full professor. This was VD, and then it was Vici, and finally he's an ERC grant awardee, and he has a very respectful, very strong, and very eclectic group in the Vrije Universiteit Amsterdam, and he will tell more about his specific research. Before we move to that, I want to tell about our position as Confocal NL in fluorescence microscopy and in application of fluorescence microscopy in protein biology and in cell biology. So, speaking of research, cell biology or protein biology, one has the options of using a number of techniques to visualize the effect in the cell, but any technique fits within a certain trade-off. Meaning, on the left-hand side in the top, you have the highest resolution method that can provide the highest resolution imaging. However, those methods are at the same time most phototoxic and killing cells. If you go to the techniques that use lowest laser powers and the most photogentle, like light sheet microscopy, those come with the trade-off of very low resolution. In the blue here, in this graph, you see the techniques that are actually confocal based technique. What I will be telling you about is our systems, which are RCM and NL5 plus techniques, which are both confocal microscopy technique, but unlike others, these are rescan confocal techniques, and they can either provide higher resolution in the case of RCM or the higher speed in the case of NL5+, but at the same time, they both work under very low laser power conditions. And the concept of the technology is when it's compared to the standard confocal microscopy as shown here, what you do see is a pinhole which serves as for the removal of out of focus blur. However, in the RCM technique or in rescanning confocal technology, we remove the PMT, and after the pinhole, we put another relay lens and then a rescanner, and then it's directly imaged on the camera chip, meaning that we increase the quantum efficiency, where with PMT, your quantum efficiency is in the range of 30%. With the latest cameras that we are using, our quantum efficiency is 96%. This is how it looks like. So not only that we at the same time increase the sensitivity of the detection compared to standard confocal, we also, in the case of RCM, increase the spot, and later we based on that we increase the resolution of 40%, going in the realm of super resolution, that is up to 120 nanometer resolution. So what does it have an effect on directly on the cells, on the imaging? We actually can, with the rescanning confocal microscopy, we can obtain brighter images even with dim signals, and even with the lower laser power, just because of the fact we have a bigger pinhole, allowing us to image and obtain better resolution than with the standard confocal with a smaller pinhole. And here, this is shown in two videos that we have. So we currently offer two types of, confocal microscopy. One is a point scanning confocal, similar to standard confocal point scanner, laser scanning confocal microscopes. And the other thing is a line rescanning confocal. In this case, with the line rescanner, we use, instead of a pinhole, like a standard pinhole, we use a slit, which allows us to image much faster, and even in the video rate of 75 frames per second. So here are the shown, just presented how our microscopes look like. On the left-hand side, again, point rescan confocal, specialized in super resolution. On the right-hand side is line rescan confocal, specialized in imaging highly dynamic events within the cell. Speaking of the first one, RCM, its hallmarks are that it can image in super resolution, so up to 120 nanometers, using nanowatts of powers. At the same time, it can be used as universal add-on to any wide field body, and also it uses a 40x magnification objectives to obtain super resolution images. If you have been doing super resolution imaging, you know that the super resolution is standardly done with 100x. However, with 40x, we obtain the same resolution at the same time obtaining a larger field of view. And here is another example how, thanks to this large field of view, one can actually image events over a longer period of time. Here is several thousands of frames. And then upon that, one can actually choose different segments within this experiment to evaluate or investigate in a greater detail. If someone, for example, wants to observe the signal transport along the cytoskeleton, one can observe it just selecting a number within those large number of frames and observe the events. Here in the case of a retrograde transport of the cargo, just by knowing the parameters and the frame times, one can actually calculate the speed of the cargo. The same goes here for mitochondria when we are talking about mitochondrial trafficking. However, mitochondria is not just that. Mitochondria, next to it being also the power plant of the cell, one of its hallmarks are the constant fusion and fission events. And one can observe, when observing over a longer period of time inside of the cell, one can actually see those events. And using this combination in combination with various mutants, for example, of mitofusions, one can go and pinpoint the exact mechanism of the event on the subcellular level. When we are talking last year, we have developed an LF5 line scan and rescanning confocal, and this year we have released on foam NL. It's the latest version. It's called NL5 Plus, which provides a high-speed imaging of 75 frames per second in a standard confocal resolution. But this has proven to be an excellent tool to image organoids. So this is an organoid image in the depth of 280 micrometers. Our latest imaging has shown that we are actually limited by the working distance of the objective. So the microscope itself can provide imaging as deep as possible. Another thing that we use next to organoids, which are perfect model for studying cancer, for example, or different types of diseases, we use it in embryology. Here is the image of 140 micrometer depth image of the mouse embryo. Next to it, there is a number more of the applications we use it. Next to organoids, we are also it is also used in embryology, like on the upper right side, observing zebrafish development next to organoids. And also we can observe very fast events. Here, what you do see is this calcium release in the root of Arabidopsis thaliana. So before I move to we move to Erwin and his work in protein biology and imaging, I want to just leave you with this take-home message. What we do is we have a unique combination of the rescan confocal technology, open pinhole to attract, because we collect more lights than a standard confocal microscope, and in combination with camera-based detection of high quantum efficiency, we can provide deepest and gentlest imaging conditions among the confocals. It's only thing upon the researchers to choose whether they want to go deeper into the subcellular level and chase for super resolution, or they are going to observe organoids or different model system that require fast imaging of the dynamic events. So thank you very much. And I would like now to give the floor to Erwin Peterman, full professor at Vrije Universiteit Amsterdam. Thank you.
Thank you, Dusan. Can you stop sharing your screen? Then I can, yeah.
Yes, thank you.
Let's see. Here we are. I hope you can see it. Yes, then let's get started. So I would like to tell you something about the research we have been doing in the lab over the past years on in vivo imaging in C. elegans, focusing on the chemosensory cilia. And I will end, most of the imaging was done with wide field imaging, but we ran into some limitations. And I will explain that to you. And we recently got NL5 Plus, and I will show you some of the first images we have taken with that at the end. Okay, we are interested in understanding how animals sense what is going on in their environment. And as many of you with a biology background know, almost all eukaryotic cells have a cilium, which you can see as the antenna of the cell. And the cilium is basically a couple of micrometer long extension of the cell that really is involved in sensing all sort of information in the environment. The model system we use to study chemosensory or sensory cilia is C. elegans, shown here. Why do we do that? Because C. elegans is easy for us to, relatively easy for us to grow and maintain, and to also manipulate to make it fluorescent. But the major advantage of C. elegans is that it is transparent, so it's really amenable to high resolution imaging. And yeah, there's no scale bar here, but for people who do not know, C. elegans is about 1 millimeter long and about 100 micrometer in diameter. Okay, C. elegans not all cells in C. elegans are ciliated, but some of its neurons are. It has 302 neurons. Each animal has exactly 302 neurons. And of these, about 50 or 60 are ciliated and are involved in sensing the environment. We focus on the chemosensory cilia, and there's basically two bunches, a couple called the amphithelia around the mouth, and I will come back to that later. But we mostly focus on the cilia in the tail of the worm, where there is two pairs of so-called phasmid cilia. And these cilia have a very interesting structure, namely that they are basically extensions of the cell membrane, and the core of the cilium is really a so-called exoneme, a bundle of microtubules. So they are really only a couple of 100 nanometers in diameter, and in C. elegans they are 8 micrometers long. What is important to note is that in C. elegans, the cilia we study are extensions of the dendrite, so not of the soma of the cell, but of the dendrite of these neurons. And what is really interesting about almost all cilia is that there is this so-called transition zone in which multiple proteins connect the microtubules to the membrane and form a meshwork that really segregates from the proteins that are in the cilium from the ones that are outside of the cilium. And not only the cytoplasm and cilioplasm, but also the membranes. The membrane compositions are really completely different. And that is really an essential hallmark of cilia. If you zoom in a little bit more in these C. elegans chemosensory cilia, they're bipartite in the sense that they have a region where the microtubules are so-called doublets, which we call the proximal segment. So here's the base, here's the transition zone. This part, this first half, sort of 4 or 5 micrometers, is the proximal segment. And extending from that are microtubule singlets, and that form the distal segment. And then in the cilia we look at, it's only the tip that is in contact with the outside world. So that needs to sense, their stuff needs to be sensed what is going on. And one of the amazing things, and that's why we got into cilia in the first place, is that there is this sort of conveyor belt, continuous transport mechanism going on in the cilia that transports stuff in the membranes, but also just components that build the cilia. And it transports stuff from base to tip and tip to base, and it goes on and on and on. And in C. elegans, in the C. elegans chemosensory cilia, it's 3 motor proteins that drive this transport. 2 kinesins, kinesin 2 and OSM3, that take care of the transport from base to tip, and IFT-dynein that takes care of the transport in the other direction. And the amazing thing of this transport mechanism, which is widely conserved over all animals, is that it sort of is not vesicle-based as dendritic or exonemal transport, but it is based on so-called trains, protein trains. So it consists of these multiple copies, hundreds of copies of these IFT proteins, which are basically, I always envision it as you have a cargo train where there is the engines, the motor proteins, but there is these carts in which the cargo to which is loaded, and that you could envision the IFT proteins like that. What happens is that at the base, these trains assemble, and it's mostly that, at least that was what was thought, kinesin 2, a heteromeric motor protein that drives the transport into the proximal segment. Then for some reason, kinesin 2 was believed to leave the trains, and I show here basically 2 of these protein complexes, IFT-A and IFT-B, but in reality it's 100 of them or so that are connected. And then sort of at this point, OSM3 takes over, was believed to take over, and is necessary to drive transport to the ciliary tip. There magic happens, and IFT-dynein takes over and transports all the stuff back again while picking up the motor proteins and delivering them back. Now, we had many questions how this cooperation of these motor proteins, how that actually works, and also how this is connected to the function of these cilia. So IFT is absolutely required for building, maintenance, and functioning of the cilia. And I will address two things that we have been studying over the past years. One is motor cooperation. How do these two kinesins cooperate, and why do we need to? And a question we have started to ask relatively recently, are IFT and ciliary function connected? So what is our approach? What has been approached basically up to now is to really use endogenous expression of fluorescent proteins in presently tagged proteins in the worms. So originally using so-called MOSCI protocol, but more recently we're switching, of course, also to CRISPR-Cas9. And that makes it, in fact, we are often studying only in the whole cilium a couple of hundred proteins at a time. So fluorescence levels are generally pretty low, but we can manage. We have so far been using, focusing on the cilia in the tail of the worm, mostly epifluorescence microscopy. And that works, as I will show you, pretty good for this particular application. But there is a cost you pay, and I will come back to that later. And Confocal, of course, Confocal and Confocal.nl come to the rescue, I hope. And we also invested enormous amounts of effort and time in data analysis, in quantitative data analysis. We really see fluorescence microscopy not as a means of making beautiful images, but as a quantitative method to really get into the numbers of what is going on. That's what we said. So we use automatic chymograph analysis, and what I will show you later, we also can do by using photobleaching. We can go to the single molecule regime and track individual motors and other proteins and really get a lot of information from that. Okay, so once you have made a worm, you throw it in your microscope. What is it what you see? Here you see an example of one of these strain proteins that is labeled IFTB, one of the IFTB proteins, and this is what you see. Scale bar of, I guess, 1 micrometer. This is the base, this is the tip. Basically, it's two cilia that are coming together and coming too close for us to resolve. And this is, again, natural expression levels of this IFTB protein. And you basically see that stuff is moving, but it's a bit of a hazy cloud. And that is not because of our image, that our image is not sharp enough, but it's just a little bit too busy to really see what's going on. So what we have been doing is we have made chymographs, time, space plots. So what you see here is the fluorescence intensity plotted as a function of time, so of image and of position along the cilium. So we really project the data on one axis, which is the ciliary axis. And we have we play some tricks with Fourier filtering, such that we can color transport from base to tip here in green. We call it antegrade transport, and transport back in red. It's measured with the same dye. It's really just a trick in the image analysis. And these are the trajectories we get. And we can do a lot with this, because here you can sort of separate different lines. We can extract them from the chymographs, and we can do all sorts of measurements on them. For example, what is shown here, the average velocity averaged over many of these strains as a function of position. And we see that in the beginning, the velocity is lower. And then after 1.5 micrometer, it really starts to increase, reaching sort of the maximum of 1.2, 1.4 micrometers per second. And you can also see that directly in the chymograph. In the beginning, the slope is a bit steeper, and it later on falls off. And so what you see is that these trains, they start moving slowly and then they accelerate, and the intensity more or less seems to stay in constant. We can also measure that. So next, what we did is we looked at the two kinesin motor proteins. And here you see the color here is really two different fluorophores, because this is a strain we made where we have the two kinesins, kinesin 2 and ozone 3, labeled with different colors. And you see, very beautifully that these sort of a single line in a chymograph turns from almost completely green, gradually here, magenta, and in the end it's only magenta. So if we average that over many worms, many cilia, many tracks in these cilia, we can get the average intensity profiles, and we can calibrate this and determine what the fluorescence intensity tells us, how many motors are connected to one train. And what we see is that a train takes off with about 50 kinesin 2s at about 10 ozone 3s. And then after leaving the transition zone, the kinesin 2 starts to fall off within a few micrometers, while ozone 3, the number of ozone 3 start to increase, reaching a maximum of about 40 per train. So what this shows is that kinesin 2 gradually hands over IFT cargo to ozone 3. But the question is, of course, what is the underlying mechanism? What is going on? And the only way, I mean, I come from the Murner lab, so single molecule is a must. And I think it really helped us to really unravel what is going on. So what we used some tricks. We either use photoactivation with photoactivatable GFP proteins, but in most cases, in fact, we use photobleaching. We are dealing with relatively small numbers of proteins. So just photobleaching 95%, we're already at the single molecule level, and we can track individual motors. And I show you a few images here. And I mean, these images are noisy, not super beautiful, but if you process them with the right software, you can track individual proteins, and that's shown here. These red circles are tracked by tracking software, and we can get individual trajectories out. And these two that are highlighted here are, in my view, already really, really interesting, because we show here that the kinesin 2 motor makes a U-turn. In principle, this kinesin 2 can only walk from base to tip, so from here to there. But apparently these motors can turn around and move backwards. What we think is happening, I will show you later on that is indeed the case, is that they fall off a train, diffuse around a little bit, and the train moving in the other direction passes by, and they bind again, and they are transported back, driven by IFT dyning. For the other motor, ozone 3, we see something similar, but of course over a much longer length of the cilia. So we can extract these trajectories, and we can do all sort of analysis. And I show just an example here, where we looked at the locations of these turnaround events, where the direction of the motors changes, and we can pinpoint where these take place in the cilia. And basically what we see is that kinesin 2 really cycles from base up to a few micrometers within the cilium, while ozone 3 really cycles over the whole length of the cilium. Of course, what we really wanted to know is what happens during this turnaround, this driver to passenger switch. So what we did is we developed tools. We wanted to do two-color single molecule imaging, but that never really worked because we use a stochastic way of photobleaching molecules, so that you then have two single molecules on one train. It, of course, never ever happens. So what we did instead is that we did two-color imaging with one color at the single molecule level and the other color just at the bulk level. So we look at single trains at the single molecule level, in this case with Cap1 and Cherry, and we look at trains at the multi-molecule level, in this case by using the IFT dynein as a label. And these are some images. It's maybe a little bit complicated. So what you see here is a single molecule trajectory of this Cap1 in white, zoomed in here, and in the background you see trains So basically all trains labeled by IFT dynein, moving forward and backward. And this again is this color scheme where we use anterograde forward moving towards the tip in red and retrograde to the back. And what you basically see in this example is that the forward motion indeed overlaps with the train. The forward motion of this CAP1 M Cherry overlaps with a forward moving train, while after the turnaround it moves in the opposite direction, and there it overlaps with a train that is moving in the retrograde direction. We could do all sort of analysis on that. We used, for example, the Manders coefficient, named after Erik Manders, the founder of Confocal NL, to indeed show that there is this absolute correlation between the motion of an individual motor protein and the motion of whole trains. So what this shows us is that kinesin 2 is almost always attached to a train. When it's moving in the anterograde direction, it's a driver, and then when it's moving in the retrograde, it is a passenger. We also did some experiments with higher time resolution. I don't have the time to show you, but there we showed that during this turnaround, in fact, there is a short moment that the motor is diffusing around. So what happens? The motor is moving attached to a train in a directed way. It falls off the train, it diffuses around until it meets another train, and then it's transported back again. Good. So this gives us a little bit of an idea of how we think that IFT in C. elegans and possibly also in many other organisms works. We have these trains that are transported mostly by kinesin 2 in the beginning of the cilium, but then we get this gradual handover from kinesin 2 to OSM3. OSM3 really takes care of the transport in the distal segment towards the tip. There magic happens, and IFT dynein is activated, and IFT dynein takes trains back and takes also the kinesin motor proteins back. And this results in a sort of distribution of the concentrations that we in fact also observe in reality. And what is important here is that what it looks like is that the backbones, the train backbone, moves in one go from base to tip and from tip to base again, but it's really the motors that dock on and off and determine velocity and directionality. Okay, I wanted to tell something else, and I hope I will manage. And that is, does IFT respond to the sensing by cilia? Why is this? These phasmid cilia, they contain the sensory machinery for chemorepulsion. Example here, we pipette something, I don't know, the worm doesn't like on its face, and what you see is that the worm turns around and make this beautiful so-called omega turn here. So elegant. So it really reacts on chemicals you supply to it. How does it do that? It does that via neuronal activity. So what we did is we tried to really image this in a nice way. We made a microfluidic chip where we can accurately dose and time different chemicals. And we looked at the response to some typical reagents. So we used hyperosmotic stimuli, and here I show glycerol, where we get, and what you see here is the calcium response of the particular cell, of the phasmid cells. And you see that we give a stimulus of about 40 seconds. What we get is high calcium fluorescence, high fluorescence of the calcium sensor, which means higher calcium concentration, and that stops after we release the chemical stimulus. And in case of SDS or other chemical stimuli, for example, copper, we see a slightly different signal. In the average, it doesn't look so different. But if you look at individual trajectories, you see that in most cases, we get a pretty sharp single peak. But in some of the cases, we get a secondary short peak. So that looks in this tailed structure. So the response to these different stimuli is different. What we next ask is whether IFT also reacts to these different stimuli. Again, I said that calcium signaling is that sets the whole movement of the worm into motion. Sorry, the calcium spikes in the neurons, that's what sets the motion of the worm in motion. But does IFT respond as well? And you see the answer here. So this is a worm that expresses, again, these two kinesin motors in different colors. And what you see is really a massive redistribution of both motor proteins when we apply the stimulus. I think the stimulus is now on, and you see accumulation of both kinesins at the tip up here. And then when the stimulus goes, you get a wave back. So what's going on? We did a lot of analysis with chymographs and different components, kinesin 2, OSM3, IFT-dynein, IFT-B. And basically what we see for all these components is that they accumulate at the tip. Kinesin 2, which is normally restricted really to the first few micrometers of the cilium, starts moving towards the end of this, towards the ciliary tip. Amazing. And if we enter stimulus, we get a very fast recovery of stuff moving back to the base. And we get similar results for glycerol or for high salt. So this really seems to be the hyperosmotic response. We asked the question, does this have any effect on the ciliary structure? And basically, the answer is no. So here we look at TBB4, tubulin, which stains the exoneme. And you see before stimulus, after stimulus. Here you see a chymograph. Basically, nothing happens. We also looked at OCR2, which is a membrane protein that we had shown before, accumulates at the ciliary tip. And in fact, we see that it increases a little bit the accumulation of this protein at the tip. And this is recovered to some extent after we stop providing the stimulus. So overall, no big changes in ciliary structure. So what happens for these other chemicals? So we use, for example, SDS, and here you see some chymographs. In fact, what we saw is that for these chemical repellents, as we call them, SDS or copper, we see much more variability in time scale and stuff of the reaction. But what we see is completely the opposite of what we have shown before. We see, namely, that the IFT machinery accumulates towards the ciliary base. Here you see that after a few seconds, stuff gets away from the cilium and really accumulates at the base. In some cases here we see the same for OSM3, but also part of the motor proteins stay behind. And we see the same for IFT-dynein and IFT-B. So stuff is transported back to the base. Redistribution often takes place in steps. First, from the distal tip to the proximal segment, and in some cases, really back to the base. Similar response for copper and SDS. We also asked ourselves the question, what happens to ciliary structure? And here we saw, we got a great surprise, because if we looked at TBB4, we really see that TBB4 is depleted from the distal part of the cilium. So the ciliary structure is really affected, the exoneme collapses. For OCR2, this membrane protein, the image is a little bit less clear. Let me come back to that a little bit later. For copper, we saw the same thing. We saw that the distal segment disappears when we apply copper. But we also saw for some reason, we obtained experiments we could image for very long, about an hour. And we could see that on the time scale of about an hour, that this exoneme regrows again, and that the structure becomes sort of normal again. You see here the cilium extending to the full length, comparable to what we had at TS0. So what is this all good for? Yeah, we wondered maybe this has an effect on neuronal activity. And indeed, if we give repeated SDS stimuli, so here we give an SDS stimulus, then we pause for a while and we give a next one. So here, this is long enough to be sure that the cilium is affected. We really see that there is, in most cases, no response to the second stimulus. For copper, we see exactly the same. So we wondered whether this could be some sort of a switching off of the ciliary neuronal activity. So some sort of habituation-like aspect. In fact, to really show that this is the case, we again waited for about an hour to see if the signal would recover. And in fact, it does recover after on the timescale of about an hour. For glycerol, the situation is different. There, if we give repeated stimuli, the cilium remains active. So no habituation there. As expected, because the ciliary structure is still intact. Okay. Yeah, one last question we asked, what happens to the membrane? And I showed you some OCR2 imaging. We couldn't really see what's happening there. So we looked at some other proteins, and here in this movie you really see very clearly what happens is that stuff is excreted. Here you see it, you see little blobs, bloop, 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 disappear like a tube of, how to call it, toothpaste, squeezed out. So we looked at some more components, and here we give some inverse color images, and you really see that we see this extracellular vesicle formation. We see that not only for OSM3, we also see it for XBOX1 and for the other IFT components. We also looked at membrane proteins, and in fact, for OCR2, we see that that is also excreted in these extracellular vesicles. We looked at TSP6, tetraspanin 6, which is a protein that people have has been labeled before as a marker for extracellular vesicles. It's a protein that seems to, membrane protein that seems to like highly curved membrane areas, and it's highly enriched in the cilium. Yeah, it really almost shows that the membrane fragments into small blobs, some of which are excreted. Okay, let me wrap up what I have shown here. So I've shown you that these neurons respond in a different way to the stimuli. Hyperosmotic stimuli, we get accumulation of the components at the tip, but the cilium still remains intact and active. While these chemical stimuli, we get different neuronal response. We see shrinkage of the axoneme, and we see this shedding of the membrane, and we might have something to do with switching off the sensory function of the cilium. So what is next? And that's where Confocal.nl comes in. The worm has many chemosensory organs. 4 of them are located in the tail. Those are the ones we have studied so far. But in fact, the ones in the head of the worm, and there are sort of 20 of them, are a bit more interesting because they're also for attractive chemosensing, and the biology is a little bit better understood there. So we really would like to image there, but we cannot because it's a really sort of three-dimensional pyramidal structure that is not possible to be imaged with our wide field techniques. We also want to look deeper into the whole neuron to look better at the soma and at the dendrite and maybe even the axon. That's also deeper in the worm. It's not in one plane anymore, so we cannot use wide field imaging anymore. It's really, epi wide field is cool if you only have fluorescence within a, how to call it, 0.5 micrometer or so of your sample. And that is what we have in the cilia, but not elsewhere. We were lucky enough to have that in the cilia. So we really need a method for these things to, with better out of focus background suppression. And for that, we recently purchased a fluorescence system of course, you need a fast and very highly sensitive system. And we purchased the NL5 Plus. And I will just show you a few images very early on. So this is the instrument in our lab, beautiful orange box. These are some first images. So this is a maximum projection of all the neuronal somas in the head of the worm. And you see that it's a whole zoo. This is the brain, the CPU of the worm. And we now can image it and we can see all the cells, and that's really cool, and discriminate them. And I want to show one movie. So this is a cilium base tip, but now we have the whole neuron, at least no, not the axon, but the dendrite and the cell body in focus. And we can image it for a very long time. The movie, what you see here, is about half an hour. And we are looking at transport of one of the proteins we're interested in, TSP6. And we can follow it, and you see there's hardly any photobleaching. So we're really super excited about that. This is a chymograph of the motion, and you see a lot of stuff happening, stuff being stuck, stuff being moving for certain amounts of time. And we are really looking into this right now, and very excited about it. Okay, let me wrap up. I'm running over time. I would like to thank the people who did the motor work, Bram, Pierre, Felix, and Andy, mostly. Christina and Guus did the microfluidics work. Of course, I should thank the rest of my lab who did many other cool stuff. Strains we get from CGC and from some other collaborators. And NWO and ERC have paid the bills. And I would like to thank you for your attention.
Well, thank you very much for this talk. Actually, I have to say that not only I was amazed by the results, but I think it was really wrapped to fit the audience. I think it was wonderful. So thank you again for really both of you. Now that we have approximately 10 minutes left, if people in the audience would like to ask question, so either you raise your hand or you can type your question within the chat. If you raise your hand, remind that if you do not want your face to appear on the video on YouTube later on, do not forget to turn off your video. I think both our speakers will be happy to take questions if you have some. Don't be shy.
So maybe I can start with the question for Erwin. So you said that on the first part, when you would like to see the individual molecules, the motors, you prefer to do by FRAP than by photo activation or photo conversion. Can you maybe comment this?
Yeah, so I should say in our case that has worked generally better, and that's for several reasons. One reason is that EGFP is a better fluorophore than PAGFP we used, so that's more every photon counts. So that is important. We have tried them, but we never really got it optimally working. So we have mostly focused on photoactivatable GFP, and there we sort of had the problem that if you use 488 or 491 nanometer for photobleaching or for activation, for photobleaching or for just detection, you activate it with that as well. So it is really finicky to get into the right excitation regime to get enough fluorophores being excited and have enough fluorescence signal. And that is just easier with photobleaching. Is in our hands for this particular system. Yeah.
Okay, and it's not toxic for the worm when you bleach like this, or?
Yeah, that's a very good question. We thought it was not, but you have to be careful. So in fact, the worm, it's not completely understood how it works, but the worm detects blue and UV light, and it sometimes also reacts on that. So we have to keep that to a limit. But I mean, I should imagine that it's important to realize that the intensities we use to do single molecule imaging are already very high. I mean, the photo bleaching is only a little bit extra. So the single molecule imaging itself might already be causing some effect. So we really have to make sure that that is okay and works fine. Yeah.
Thank you.
Okay, so I may have questions as well. So both some of them related more on the biological mechanisms, and some others maybe on the more on the technological side. So at first, it's actually it was very unknown for me to know that the motors and the cargo, everything can reverse in order to move back, and so. But you were very elusive in the mechanism of the reversing. So they detach, then they diffuse, and they reattach, but by chance on the other way around. Do you have any idea of that, how that could happen? Are there any sort of chaperone or wrapping molecules that favor that movement, or
so
how does it work?
What is how to call it? I wanted to tell too many things, so I sorry for missing some of the detail.
No, come on. Yeah.
Very important to realize is that so the direction is set in a way by the microtubules. They're all pointed with their plus end outward. The kinesins only move from minus end to plus end. The dyneins only from plus end to minus end. What we think is going on is that as soon as the motors detach, and that's the same for dynein as definitely for the kinesins, as soon as they detach, they go into an inactivated form and cannot bind to the microtubules.
Okay.
Only binding to cargo can result in their activation. That is, there is quite a body of evidence for that, for many different kinesins, also for these. So what we think is really that this binding affinity is subtly organized, and that might be location dependent. It might be different for anterograde moving trains to retrograde moving trains, most likely. But the details we do not really understand. But it's we're sure that we only get directed transport either as cargo of a retrograde moving train or as driver of an anterograde moving train when the motors are bound to a train.
But is there any modification of the protein, like phosphorylation or this kind of things? Or
yeah, that's a very good question. People have investigated, and we are also investigating these kind of things. We have not really been able to pinpoint phosphorylation, and post translational modification definitely play a role, but it's not a simple story that this switch on and off takes care of all the transport. So there is now an amazing body of electron microscopy data appearing, where people in situ, not in C. elegans, but in Chlamydomonas, can really image at, what is it, angstrom resolution, few angstrom resolution, whole trains, and really the idea is that the structure of trains moving forward and backward is really different. We know the structure of trains moving in the forward direction is now well understood. The structure of trains moving in the opposite direction is not yet clear, but I guess that that will be a matter of time. And how that is orchestrated, that's not yet clear, but it could also very well be that it's really the train that sets many of the aspects that define how the motors bind and if they can be active in that way.
Do I have time for one question, Gael, Virginie?
Sure, I think we can, yes.
Erwin, one thing, this whole concept looks so tightly regulated and nuanced. So did you try to manipulate the levels of kinesin and dianine?
Yeah.
Just lifting one up, lowering one down, and see what's going on with the transfer? Because I guess you would get very weird results depending on what you do, right?
We have been trying that. We never really succeeded. But how to call it? We're working on it. I just wrote a grant, etc., etc. So this is really so this is a great remark. This is exactly what you would want to do to sort of reduce the number of proteins. So almost, of course, a lot of stuff is being done on mutants where really the function of one of the motors is completely lacking, and that gives interesting insight. But in many cases, these worms are a little bit sick. So sometimes you see accumulations. It's basically, then you have sort of a train accident. You want to see the train accident happening, not the, how do you call it, the train wreck at the end. That is what we want to do. And that is exactly a way we would like to figure it out. But we haven't been able to do it so far.
Thank you.
Well, I may have one last question related to both of you. So, Erwin, you were telling us in the very end that actually the new system could enable you not only to increase the duration for which you can image the process happening, but also the field of view. And I was actually wondering if you can estimate what is this improvement in terms of not only, yes, time, but as well, how many times more have you been able to increase the field of view? For example, what was actually the objective that you were using before and the related field of view, and now the one that you are usually using now? And if you can have an idea of all the expectations in terms of improvement in both space and time that are beneficial.
So we typically use different objectives for different applications. Mm-hmm. So I would say the big difference in this case is not so much the objective; it's the camera.
Ah, yes.
So the how to call it? For all the wide field experiments we use an EMCCD camera with 512 by 512 and big pixels. But of course, for the line scan we use what is it? An FCMOS, small pixels, and many more of them. Look, we are, of course, limited or restricted by the size of the animal. So I would say, let me come back to what you said. So I think what I think is that sort of with the confocal system, we will not be able to do, or I'm pretty sure we will not be able to do single molecule imaging. Mm-hmm. But for all the other things, it might really work well. Our single molecule imaging, we are lucky to work on a system that is almost one or two-dimensional, if you would want to say. And that really, that's why wide field imaging works in that case. Otherwise, confocal will always be better. And so that's what we hope to do, to also really get three-dimensional information and these kind of things. And then, I mean, the image I showed you shows really that what Dusan was telling, the confocal instrument, this confocal instrument is very good in measuring every photon, so reducing photo bleaching a lot. And I think for live imaging, that is so important.
Okay, great. We just had a question actually in the chat from Chloe. So it's in French, so I will translate it. I think it's more dedicated to Dusan. So Chloe is asking how the Confocal.NL system does improve the resolution up to 120 nanometer in X and Y. So she said that she understood that you do that by increasing the size of the pinhole, and in turn that you can diminish the laser power. And so by, I think she's confused by how increasing the size of the pinhole, you still diminish the resolution. I think I have translated it more or less.
So, okay, I'll try to do it in a shortest possible time because I was warned by you. So these two separate things. So what we do based on the scanning and rescanning system in the animation I have shown you that we are literally stretching the spot, and then by reducing the image, we actually improve the resolution. The improvement of resolution through the rescanning confocal microscopy system, RCM system, is only for RCM, not for NL5+. In RCM, it goes for 40%. So in theory, raw resolution increase is 170 nanometers. We also have, within our system, with our RCM system plugin, we also have a deconvolution software with this microvolution that further reduces the resolution, improves the resolution from 170 to 120 nanometers. Microvolution is one of the options because it provides deconvolution on the fly. So you can do the live cell images to do the live cell super resolution. We also can do deconvolution through SVI hyphens, which uses a different type of mathematical operations, but this is happening in post-processing. For live cell super resolution, 120 nanometer, the combination is RCM2 or RCM 2.5 together with microvolution. Is this okay as an answer?
I think it is. Then it's on Chloe to, yes, she said yes. So I don't know, Virginie, maybe we can, I think if you have and all.
Yeah, exactly. I think it's time to close and to thank Dusan and Erwin.
Thank you.
And thanks to Confocal.NL to have proposed this meeting and these speakers. So thanks a lot.
Indeed. Thank you a lot. Thank you to the audience as well for being attending this talk.
Thank you.
Thank you. You're welcome. Bye.
And the next meeting, the next video conference will be the 5th of September. So we do a break in August. So next meeting, the 5th of September, and it will be with Andor company. So thanks a lot to you all, and have a nice summer. Bye. Thank you
very much.
Bye.